All right. Well, people are filtering in on this Easter holiday weekend. We have a light crowd, but a very important crowd. We're going to talk this morning about our series we're going to be doing over the next several weeks, reviewing new data from the Conference on Retroviruses and Opportunistic Infections. And so the first piece of that series is being presented by Stephen Rawlings. Stephen is one of our wonderful infectious disease fellows here. He did his medical school training and received a PhD in HIV immunology from NYU in New York, and then came to us here in San Diego to do his internal medicine training. He's been in the physician scientist training program and that track, and came to us here in our infectious disease fellowship training program 2 years ago. Stephen is working in the Richmond and Smith lab uptown on HIV-related research this year after completing his clinical year of training. And he's going to review therapeutics and cure research as presented at this year's CROI. So Stephen, welcome.
Thank you very much, Connie. And if there's any mic issues, let me know, but I think we're streaming. We're also trying Zoom for the first time. So I have no disclosures. A brief outline. There was a lot presented at CROI, and it was all very interesting. I tried to synthesize mostly talks from the morning of the first full day, which was dedicated to cure. Some outstanding oral talks that I think show a lot of progress, but also some work still to be done in the cure field. So we'll talk about trying to reproduce the Berlin patient, the one patient known before this meeting to have been cured of HIV, other genetic modifications that are being sought for curing HIV therapeutic vaccines, some other antibody work, and then shock and kill versus block and lock. So the first one was a case, the London patient, which actually got a lot of news. A second person, we're calling it remission so far. But this was a talk presented by Dr. Gupta from the University College London. And actually they simultaneously managed to get it published in Nature and had a nice news release at the same time. So he started with talking about the original Berlin patient, which I think most of us are aware of, but basically Timothy Ray Brown was living in Berlin, an American living in Berlin. He had been diagnosed with HIV many years before developing AML and required 2 rounds of chemo and stem cell transplantation, but there was a twist added to his stem cell transplantation. Instead of using just HLA-matched donor cells, the German registry also had information on this CCR5 delta 32 mutant allele, which in homozygous individuals actually protects against acquisition of HIV. Timothy Ray Brown had what seemed to be a CCR5 tropic virus, and so when he was given essentially cells from this donor that didn't have the CCR5 receptor, he was able to go off of his antiretroviral therapy and did not experience rebound viremia. And this was really promising 10 years ago. And everyone thought that we'll start doing this again or in more individuals and it'll work. And it hasn't worked. So basically there was you start to learn a lot about the geography of Europe, but there was an Essen patient that had escape of an X4 tropic virus. A couple cohorts or a couple of individuals in a cohort in Philadelphia were given heterozygous CCR5 delta 32 stem cell transplantations, and they had viral relapse. And so there was a thought that maybe either we were missing something or maybe, the amount of treatment that the Berlin patient went through, there were 2 rounds of total body irradiation, an aggressive destruction of his normal immune system, and then some graft-versus-host disease from the transplanted cells, perhaps things that really have high morbidity and mortality anyway. He fortunately has survived, but perhaps we just can't repeat that. And so what Dr. Gupta presented was a case of a man living in London who had had diagnosis of HIV in 2003. When he was initially diagnosed, actually, he wasn't started on any antiretroviral therapy. Um, 10 years later, in 2013, he was diagnosed with stage 4B Hodgkin lymphoma and was initiated on antiretroviral therapy with Atripla. But they were having trouble treating the Hodgkin, and they switched him over to an integrase-based regimen because he was going to get chemo. Multiple lines of chemotherapy failed. He was unable to mobilize cells for an autologous stem cell transplant, and so they began a search for a non-related donor for allogeneic stem cell transplantation. They found an unrelated 9 out of 10 HLA match that was homozygous for CCR5 delta 32 mutation, similar to the Berlin patient, but they were not planning as aggressive a conditioning regimen. So he got lomistine, cyclophosphamide, cytarabine, and etoposide, no total body irradiation. And his course, that was his conditioning regimen. He was then given the stem cell infusion on day 0. His post-transfusion course was complicated, as often happens with our bone marrow transplant patients, with gram-negative sepsis. He had a dental abscess. 77 days after transplant, he had colitis, and biopsies taken of upper and lower gut suggested possible graft-versus-host disease. He also suffered CMV and EBV reactivation, which were treated, but did quite well overall. And so here's his course. The viral load, HIV viral load, is in blue, and the CD4 count is in red. Shortly before transplant, which is here day 0, there was a 5-day interruption of his TDF, FTC, and raltegravir. And actually, in that course, he became viremic. They checked it, confirmed it was an R5 tropic virus, but they also discovered what had not been present previously, an M184V and K65R mutation. So they switched him over to a ripivirine, lamivudine, and dolutegravir regimen, and he was suppressed. 18 months in, his antiretrovirals were stopped, and he has not had any rebound viremia since. I'll show you in this slide. They look here. So the orange is chimerism with his own cells here at time point 0 before transplant, and then the donor cells engrafting what appears to be 100% after the transplant. And then looking at CD4 or HIV in cells before transplant and afterwards, also with a series of QVOA assays, has been negative. Here, after, I think this is about 12 months after stopping ARTs, they did a number of high sensitivity assays with leukapheresis, couldn't find any, sorry, I don't think it was leukapheresis, but in large blood cells draws, they couldn't find any CD4 by LTR or GAG copies. They did do this one screening where 1 out of 8 was a low-level positive for LTR here. But otherwise, no evidence of rebound virus. And so they did a little bit of comparison to Timothy Ray Brown, what we're calling the London patient. So the London patient was actually homozygous for CCR5 before going into transplant. Timothy Ray Brown had actually been heterozygous. Both appeared to have an R5 tropic virus before transplant. Their malignancies were different. The amount of stem cell transplantation was decreased in the London patient. The conditioning was less. They both underwent a T-cell depletion, but it was with CD52 in the London patient. Both had some graft-versus-host disease, and both exhibited high, what appears to be at least complete donor chimerism. That did come up in the question, so I'll talk a little bit about the discussion that happened afterwards and my own thoughts. So one of the questions, the presenter, Dr. Gupta, was very careful not to say that this was cure. He was calling it remission. There were a couple journalists from different publications there. The journalist from PAUSE came up and said, well, when do we get to call it a cure? And I think that's a reasonable question that the field is still struggling with, because we've thought that people were aviremic before and then just had rebound much, much later, weeks to months after an announcement was made. I think Mississippi Baby's an example where we thought that was a cure, and then was it 21, 23 months after she was lost to follow-up, represented with viremia. So his argument was, well, 18 months is pretty long, but we're going to keep following, and they're going to still say remission for now. Another question was actually about the chimerism. So he's saying it's 100% donor chimerism, but that's based on their clinical assays, which I believe is a flow assay. If they are planning, it sounds like, more sensitive DNA sequencing assays, but they hadn't done them yet. They did check for surreptitious use of ART, which has been an issue in some of these analytical treatment interruption studies, but on 2 separate occasions, he was not surreptitiously taking antiretrovirals. They had not yet assessed any virus in the CSF, which is, and this was also talked about in some other presentations, but perhaps a privileged site that could have different characteristics or kinetics of rebound. But they were planning to get a lumbar puncture, and a fair argument too was, even if there's some rebound occurring in the CSF, presumably it enters the plasma again within, we don't know the total time scale, but weeks to months, and he's so far been negative. And I think, just this gets back to that question that I was bringing up at the beginning, where we still don't seem to fully understand what is needed to replicate this clinically. So I mentioned Essen patient where similar transplant, but there was X4 tropic virus that grew out. So certainly this is only going to work if you don't have any archived X4 tropic virus, or if you have archived X4 tropic virus, but we managed to eradicate it with enough conditioning, perhaps then it would work, but it's not clear what conditioning is still necessary and so on. So lots of open questions, but promising from the standpoint that at least has been replicated. So I want to move on then to another concept for reducing CCR5 expression as sort of a way to reproduce the Berlin patient. So this is a lot of work out of Carl June's lab at UPenn. Pablo Tebas presented this talk. And so they've been playing around for a long time with zinc finger nucleases, which were the cool thing on the street before CRISPR-Cas. But basically, an adenovirus can encode these zinc finger nucleases that will go in and cut out enough of the gene for CCR5 to create a CCR5 delta 32 equivalent T cell. They apherese blood from donors, enrich CD4s, treat the CD4s with their virus. They look for something around 13 to 36% cells with both copies knocked out of CCR5. They expand these and then essentially reintroduce them into the donor who's also his or her own recipient. So they published a trial where there was no conditioning, no cyclophosphamide, and essentially everyone became viremic. There was some other work that they've shown too, that if you start off as a heterozygous, you at least delay your viremia, your rebound. But they thought, well, maybe we need to add some conditioning to try to make it easier for the reintroduced or transfused modified cells to expand and sort of increase their ground. So they had 2 cohorts, one with wild type CCR5 and then one that it's just 3 patients in each, but was delta 32 heterozygous. It looks like they gave them 2 doses here, but he kept saying in the talk that it was just 1, so I'm not clear why they have 2 different doses of cyclophosphamide. But essentially, everybody got cyclophosphamide 2 days before reinfusion of their modified cells. So that's what this looks like here. 10 weeks before they enroll the patients, they leukapheresis, they prepare the cells, they give the patient cyclophosphamide 2 days before an infusion of their own modified cells, monitor for 8 weeks, and then undergo analytical treatment interruption, resume. There was a plan to resume ART at 24 weeks, and then go from there. And so what they found was that everybody rebounded, although some rebounded a little bit later than others. He tried to put that all together on this slide. So one of the issues that was brought up was, and this is just something that's affecting the field, but we no longer think it's appropriate to have essentially non-intervention controls undergoing an analytical treatment interruption. So to compare the rebound viremia, they used historical controls, which is this gray line. And when they pull out their cohort 3, which were the patients that were heterozygous already for CCR5 mutation, they thought that there was a statistically p.04 significant delay in rebound viremia. The trouble is it actually only is 2 patients because one of the 3, patient 305, was surreptitiously taking ART, so that patient was excluded from analysis. This is a complicated slide, but basically the point here, they were trying to look at the prevalence of the mutated gene. They couldn't find anything statistically significant, but they were arguing that in this cohort 3, there was a trend towards significance in terms of greater amounts of the disrupted gene present in peripheral blood by deep sequencing. And then they had 3 patients. They couldn't have all been from cohort 3. I don't remember if he said which cohort they were totally from, but who actually wanted to go past the 16-week reintroduction of ART. And so that's these 3 here. And his point was just really, particularly this patient, so even 2 of them didn't get terribly high viral loads. I mean, this is 1,000, so this is, something like 9,000. But this patient in particular is out almost a year before resuming ART, and is just hovering in the mid to high thousands. So maybe there's something there, but it's still rebound. So their point was just it's safe and well tolerated, this system that they're using. They can disrupt the genes. They think that the cyclophosphamide increased engraftment compared to their prior study without cyclophosphamide. There seems to be a delay in viral rebound, but their numbers are low. And then a few individuals did have very sustained low-level viremia, and maybe we're getting closer and closer. This is certainly something that has a lot fewer side effects compared to total body irradiation or any sort of chemo conditioning. So it has some promise. And then if you really want to start getting weird, this is interesting but scary. So this is a series of experiments using CRISPR-Cas in non-human primates, but they're presumably going to attempt it in humans soon. So essentially, this is now using CRISPR-Cas, which is another way of editing genes and removing genetic material from the genome. But these are targeting sequences within the HIV integrated sequence themselves. And so they can show essentially very large deletions in integrated HIV that is lethal to the virus. So I skipped over some of it, but they were showing essentially ex vivo it works. And so they took, I forget how many animals, but they had primates that were infected with CIV. They gave them an adenovirus encoding the CRISPR-Cas. So essentially we're just introducing a new virus and hoping it's going to find all the cells that have HIV, or in this case, SIV in it, and modify the genetic information to specifically remove HIV and not harm anything else, wink, wink, nod, nod. And so it was limited what they were able to show, but show a little bit of their data. Basically, they could find that this adenovirus could introduce CRISPR-Cas in a number of tissues, essentially every tissue that they looked at. And when they mixed the PBMCs of the animals, it no longer produced virus. So I'll show you that. So here's just gels looking at the actual genetic products now in 2 animals that were treated. So you can see much smaller fragments suggesting deletion of SIV, although there are some residual full-length fragments. This is a control that didn't receive it, and you can see none of the smaller products present. Um, and so when this sounded a little shady. I suspect that maybe they're seeing some viral outgrowth, but if they don't activate PBMCs, so they just let them sit together with a permissive cell line, so peripheral blood cells taken out of these animals that were treated with the CRISPR-Cas, there is no viral rebound. So essentially all these points down here. If you have a control animal, it rebounds. But the presenter mentioned that they were undergoing further experiments with activated cells, which makes me think that they actually found viremia and they just didn't want to show it, but they're trying to figure out what's going on, which is fine. In tissue, so here's spleen, lung, and lymph nodes, animals that were treated did have successful reduction in the full-length genomes and production of these shorter genomes, but again, it's not 100%. And so that was limited data, but quite impressive. So essentially they can confirm that they have this adenovirus delivery vector that works, the CRISPR-Cas works. They can cleave the genomic DNA of SIV. They can reduce viral outgrowth from PBMCs. They can find it in the introduction of the CRISPR in multiple different tissues. And so perhaps this is an avenue for future therapy. A couple of questions came up. So they had not done a treatment interruption yet in the animals. This was more of a proof of concept of the delivery vehicle, but they are planning those. And then one of the questions I thought was a good one that somebody asked was whether we know there's a significant amount of viral diversity of SIV, HIV, and could you essentially have an escape mutation that would evade the CRISPR-Cas? And they said that they had gone very carefully through the Los Alamos database to find well-conserved regions, and they actually have multiple targets, so they thought that that wasn't the case. But I guess that still is to be seen. They'll obviously want to sequence any virus that grows out, should something grow out in an analytical treatment interruption, and see if that's how it escaped the genetic modification. Okay, so moving on. Another avenue is therapeutic vaccines. And so the idea here is using broadly neutralizing antibodies as therapies. There's a lot of interest, obviously, in inducing broadly neutralizing antibodies as prevention. But the therapeutic option here would be that we can either induce a host to produce his or her own antibodies that will control HIV, or just passively transfer, which is where most of the work's being done right now, essentially infuse a potent broadly neutralizing antibody to control viremia. And there's been publications on this already. It doesn't seem to be working completely, but we are getting prolonged rebound-free analytical treatment interruptions. So there's some signal here. But one of the concerns was that an antibody with its 2 FAB fragments might not be enough. Well, let's make 3. That sounds better. We can engineer this to be even more impressive, and we can make sure that it's not just a simple antibody with 2 of the same things. We can put 3 different sort of broadly neutralizing globs onto a molecule, and now you kind of have 3 in 1. I guess that it's a fine idea. That's what they wanted to show. It seems like a lot of work, but they've done it. So Dr. Pegu from the VRC was presenting this talk, and basically they have this trivalent, trispecific, antibody model where they have a VRC01, MPER, and 10E8V4. When they compare it to other monoclonal antibodies, its potency is at the high end and its breadth is at the high end. So they like their system. And they've shown that it seems to work very well in a series of strains of SHIV in viral escape. This is in vivo though. So what they tried to do was introduce it to SHIV model with non-human primates. And so the slides were suppressed. So this is just basically what he was saying. They had 6 animals. They gave them the infusion of the product, trispecific antibody. They saw dramatic reductions in plasma viremia, in many cases a 1,000-fold decrease, but it wasn't completely abrogated, but this was interpreted as evidence of viral control. And then he mentioned, unfortunately, even though the levels persisted for a long time after just, I think it was 2 infusions, a week apart, many of the animals started to develop anti-antibody responses, which is sort of the big Achilles heel of this sort of approach, that we're seeing essentially our own bodies attack the antibodies that we're trying to use to treat the virus. And so we start to lose efficacy. I said that actually this product is going to go into clinical trials later this year. So I think there's more to come in this arena. But his conclusions were just sort of, as I said, it binds, it seems to inhibit outgrowth of virus in vitro and in vivo, and more to come. If some of you know, I sometimes rail about other antibodies, the monoclonal antibodies, and so there was an interesting talk on PD-1 antibodies, and anyone who was at ID rounds yesterday also saw Katya talk about pembrolizumab in JML, or PML, excuse me. But so this was a study actually looking at what pembrolizumab, which is an anti-PD-1 monoclonal, can do to decrease the HIV reservoir and sort of in a form of shock and kill, actually reverse HIV latency. And so they had some data for this. Also, at least one paper in one case with a similar antibody, nivolumab, decreasing the reservoir. And so that's just to show you from that paper, from last year, they did cell-associated HIV in this patient who was receiving nivolumab and showed a decrease in this measure of reservoir, although other measures sort of stayed the same. And so Thomas Uldrich from the Hutch presented this series where they have a cohort of individuals undergoing therapy with pembrolizumab. Some of their background data just was that we know PD-1's upregulated in HIV, and essentially reversing or inhibiting the normal function. And just to remind us, so the PD-1 is thought to be something of like an inert energy-inducing signaling molecule on the surface of T cells. So a T cell goes out into the world and it says, I think I'm going to activate here, and if it has a high level of PD-1 on its cell surface, it's sort of like a brake pedal that's available to slow things down. And if a neighboring cell or a cell that the T cells involved in is expressing PD-L1, the ligand, that's essentially becomes the foot pressing on the brake. And so we know cancer cells can sometimes express this ligand, and that causes decreased immune recognition and killing of cancer cells, which is the reason these drugs work in some cancers, because essentially the antibody comes in and prevents the brake from being applied, prevents the foot from stepping on the brake. In these cases, here in HIV, this is not now cancer studies, although some of the initial observations were in patients who needed the drug for an unrelated cancer. But the question is, okay, are there reservoir cells, T cells harboring latent virus that are not becoming activated sufficiently because they're seeing too many foots pressing on the brake? And so can we inhibit that and actually cause some kick of the reservoir? And so the trial is CINTN12, a multi-center trial where they are enrolling participants who have been on ART and have a range of different CD4 cell counts. They wanted to demonstrate safety across this range. And basically, this is their enrollment table, patient demographics table, but pretty much just varying by the starting CD4 count, the baseline CD4 count. And so the interesting component, they found a statistically significant increase, so just one dose of pembrolizumab, and an increase in unspliced intracellular HIV RNA at day 8 that seems to not be significant at day 22, arguing here that you are actively kicking, if you will, the increasing the amount of virus. But then no significant increase, although it looks like maybe it's coming up, of the plasma HIV RNA. So again, asking, is there some HIV coming out? Now, when you're continuing your ART, the thought is, okay, you're not going to infect new cells, but this is the whole shock and kill idea. And so they then look for a measure of the reservoir intracellular HIV DNA, and they saw a statistically significant, but what seems like very small decrease very early that seems to go away within weeks of the infusion. And sort of going back to the unspliced, there's a increase at day 8. So there's some activation of the virus and maybe even a loss of some reservoir cells because of this application of the foot inhibitor from the break. But it's all restoring to sort of homeostasis within 3 weeks. So maybe there's something here, but it's not huge. So their conclusions, we talked about transient increase in HIV transcription, consistent with a latency reversal, and a decrease in the HIV DNA, but its ongoing long-term effects don't seem to be persisting. It's still something interesting, and this, well, we talked about a little bit yesterday too, there's some side effect profile that's not as simple as some of the other things I talked about, because there are essentially de-repression of the immune system effects where you can get sort of autoimmune type symptoms, but maybe this is safer than some of the other things being attempted. So that's sort of a shock and kill. There's one other presentation to talk about shock and kill. I don't have a lot to say about block and lock, but just to point out that's another idea here that as we understand more of the mechanisms underlying latency in terms of reversing latency, which is shock and kill, maybe we could figure out a way to actually just increase latency and in a sense lock the virus into cells. There was some talk about that I won't go into, but it's another avenue that's being pursued. So the last thing, in a series of HDAC inhibitor experiments that have been going on for at least a decade, the latest that's had some interest is romidepsin. It's in that family of varinistat HDAC inhibitors, and it seemed to work better in vitro. So a question was, how does it work in vitro? I keep switching around. How does it work in vivo? And so Dr. McMahon presented this trial. Again, a reminder, basically these HDAC inhibitors seem to be able to, at least in vitro, reverse the epigenetic modifications and suppression of HIV, which is what we're calling latency, and cause the cell to become more active. If good, effective antiretroviral therapy is in place, then no new cells won't become infected, but viral cytotoxicity will cause the latent cell to die, thereby overall decreasing the reservoir. And so romidepsin is already in use for T-cell lymphoma. So these HDAC inhibitors often come from the pipeline of chemotherapeutics for cancer, but it had worked in vitro, as I mentioned, and in a couple of studies before this in vivo, it hadn't seemed to be as effective, but they thought that the doses were too low. And so I didn't show all the slides, but basically the short of this is even at the higher dose, it still didn't really work. So they gave a higher dose of romidepsin. Generally, it was well tolerated. It's still below the maximum treatment dose. When they give it, they don't see any changes in any of their reservoir measures that were significant, but they maybe see some changes in the total activation profile of the CD4s, so maybe there's still a signal here, but I'm just still, like many, waiting for something that works this well. It works as well as the in vitro studies in vivo, and this still didn't seem to be it. And this is what the slide, in their conclusions, they mentioned the ex vivo and in vivo virologic responses just aren't connecting. So there are some trials. The thought is maybe this family of HDAC inhibitors, romidepsin being perhaps the leading candidate, with some other piece, whether it's a broadly neutralizing antibody or efforts to increase cell-dependent killing, could be the key. And so a mixture of avenues of cure is being pursued, but no updates on that yet. So my conclusions, I think that there's a lot of renewed hope just by doubling the number of cases of quote unquote cure that have been presented now. And so we are definitely not done, but it's thrilling to see that there's some progress being made. I think gene editing, if you're comfortable with it, is something that's definitely advancing and might have a lot of promise. Antibodies and these latency reversing agents don't seem to be there yet, but for the most part, they seem to be a better side effect profile, so it's worth pursuing further. And then there's still this wild, wild west of checkpoint inhibitors, where we're finding interesting things when we muck with the immune system, but it might still be best in the chemotherapeutic range for active cancer. But at the same time, if we can find something that makes it easier to eradicate the reservoir of HIV, then I think there's some promise. Just some photos. This was a really cool point at the end. This is Dr. Gupta, and this is Timothy Ray Brown. And Dr. Gupta has just told the world that Timothy Ray Brown's no longer alone. So I thought that was kind of a cool moment. It's a great front row shot of our wonderful San Diego representation, part of it in CROI, and here's another cohort of us hanging out at lunch. That's it. Thanks. I'll take any questions.
So I want you to go back to the.
I think it's on.
To the CRISPR-Cas.
Yeah.
And talk a little bit more about what you don't have to go back.
Oh, okay.
Thoughts on what's scary about it, and where you see that going.
Yeah. I think the scary part that everyone, it's more just tempering expectations on CRISPR-Cas from the standpoint of if there's off-target effects, we may not find them out. So could it possibly recognize some retroviral sequence in a retrotransposon that's in the human genome and end up cutting out a anti-oncotic gene in a cell? And I that's not my area of expertise, so I'm willing to defer to the experts, but it is the piece that everyone's always worried about with these gene editing, stem cell editing, or even this China baby with the CCR5 mutation, is not so much proving that you can do it and that the gene you wanted worked, but what about the things we aren't looking for that could still happen? So, and then what if it doesn't even totally work? They still had plenty of full-length genomic virus in their tissues, and they haven't done the study to see if it's effectively a cure with a treatment interruption. So it's exciting, and I think there's certainly reason to believe it could work from the standpoint that the sequences should be unique enough that you could find just HEV genomic sequences and just cut those out. But all it would take is one cell that's mutated in the wrong way, and you've just given someone cancer. Any other questions? Yeah.
Thanks, Marvin. I'm a little unclear as to the, it's hard for me to see the exact role in the antibodies. I know that they're nice, and I know that they're fancy, and I love the tri-specific. It's pretty cool. And I know that they're not free from,
complications.
Well, autoantibodies forming against them and stuff. But as far as kick and kill, right? I mean, we have antiretroviral therapy that can be used for the kill approach. So
I think part of it is an idea that obviously an antibody half-life in the absence of autoantibodies can be very long. So you could conceivably, just like we're looking into, depot shots of ribavirin and dolutegravir, what if you could just set someone up with a clinic infusion of a good cocktail of broadly neutralizing antibodies, and they don't have to take antiretroviral therapy for 3 months until their next infusion, because the antibody controls viremia so well that it's what we call a functional cure.
Exactly, which is part of the reason why I'm like confused, because we're already working on long-acting injectable antiretrovirals.
And then the next question is, how comfortable are you with gene manipulation? So there are systems using that adenovirus vector where you can introduce the genes necessary to make the broadly neutralizing antibodies into an individual, and their own cells become the factory. So it's being investigated certainly for vaccine efficacy, because it's hard to elicit from standard vaccination methods these broadly neutralizing antibodies. So why not just give somebody the DNA? But you could imagine, or the people doing this imagine essentially a shot with some nucleic acid transfer mechanism that ends up turning your own body into the factory, making your own antivirals. And so now you need nothing except periodic visits to ensure that your viral load is being controlled, you're making enough antibody, and you're not making too much autoantibody. So that could be promising.
Century.
Oh, sure. Sure. All of it is. I mean, we have to think outside the box. None of the standard stuff's working. So. Except antiretrovirals. All right.
No more questions? Okay. Thank you.